All right, can you all see my screen?
Yes, please.
All right. So for today we'll be treating the Paramecium, and it's one of the microscopic organisms that you'll be studying in the course of you studying active biology in high school. Why you be studying these organisms is because of the kind of features that they have that help them survive as unicellular organisms. All right. So the Paramecium is one of the very popular unicellular organisms, and it dates as far back as the 17th century, and it's believed to have been very known to Anton van Leeuwenhoek, which is the man or the inventor of the microscope. So it was one of the very first microscopic organisms, or the unicellular organisms, that were viewed under the microscope of Anton van Leeuwenhoek. Are we good?
Yes.
Okay. If you have any questions, you can post them in the comment section or raise your hand so that I see you, and then I can ask whatever question you have. Are we good? Okay. If your responses are a measure of how you are taking the class, so I would like everyone to respond where do you, and please mute your microphones if you have nothing saying. If not, I'll take you out of the meeting. Okay. So the Paramecium is known to be a protozoan protoctist. And now these protozoa, what are they? They are single-celled eukaryotes that are free living or can be parasitic, and they feed on like microorganisms and other organic tissues or debris. So when we talk of eukaryotes, you know what eukaryotes are, right? I told you the last time, you meaning true, and then the karyotes or karyon coming from the word nucleus. So true nucleus. So it means that the Paramecium is a single-celled organism that has a true nucleus, and it's single-celled, it's free living. So it doesn't have to depend on any other organism for survival or any other mechanisms for survival. As a single cell, it's able to survive on its own and carry out all the life processes. It's parasitic, meaning it could feed on other organisms for survival. It could feed on some organic matter, it could feed on debris. So these organisms are mostly found in brackish waters, ponds, etc., because these places are filled with microorganisms or probably bacteria that these Paramecia feed on. So if you want to locate Paramecia, you mostly find them in brackish waters, marine settings, ponds, stagnant waters, basins, etc. So anywhere you could find these micro or smaller living organisms to which Paramecia can feed on, or you can have plant debris or organic debris where Paramecia can feed on, there's a likelihood of you finding Paramecia there. Now, we said protoctists, right? Now, protoctists are any unicellular or simple multicellular organism belonging to the kingdom Protoctista. So further down the course, you'll be doing classification or taxonomy, and you get to meet all the kingdoms that we have. So you'll be grouping animals and studying animals according to kingdom, phylum, class, order, division, genus, species, etc. So you'll be grouping these animals there. And then we are saying that the Paramecia is found under the kingdom Protoctista. Please, is that good? Okay, so now, why would you want to study the Paramecia? As I said before, they have certain features that make them very unique, because it's quite wonderful how, as a single cell, they are able to carry out most of the life processes. So it's quite fascinating, and then scientists would like to study these organisms even as they are as single cells to find out what mechanisms they have and how they carry out their life processes. So first off, you are going to be talking about their body morphology. So if you've seen Paramecia before, you've seen images of Paramecia before, we say that they are slipper shaped because of how they look like. I have a diagram in the slide I'm sharing. I'll show it to you shortly. Paramecia look like slippers, let me say it that way. But because they are microscopic, they are mostly seen as a grayish speck, right? So if you locate, it's difficult to see them with the eye, but if you're able to locate them in the ponds, you find them as gray specks, like tiny, very tiny. But if you view them under microscopes, they are mostly slipper shaped, and the length is about 0.22 mm in length. So that's very, very small. It's lesser than 1 millimeter. So most of the time, these Paramecia are transparent, as the point 2 says, transparent body, so slipper shape with both ends blunt. So because of how transparent they are, if you should place them under a low power microscope and you want to view them, it will be difficult. So most of the time, the slides that are prepared are stained. So when you stain them, the stain affects the build up or the makeup of the paramecium, and then it makes it easier for you to see. So it's kind of like it stains the body of the paramecia so that it makes it easy for whoever is viewing the microscope to see whatever it is. So note the length 0.22 mm, slipper shaped, and it's transparent. Now it also says it has both ends blunt, but then if you look at it, one end is more tapered than the other. You see that shortly in the diagram I'll be showing to you. Now, one unique thing about the Paramecia is its body. So now it's covered in a substance known as the pellicle. And then the pellicle is made up of a thin gelatinous substance produced by the cell. So that's what the pellicle basically is. And then one notable thing of Paramecium is that they are heavily ciliated, right? So they have, if you look at them with low power microscope or even high power microscopes, you see tiny hairs all over the body. You see a lot of tiny hairs all over the body, and this is what we call the cilia. And it has been found that the cilia are made up of substances we call microtubules, right? Further down, as you are learning deeper into cells, you get to know what microtubules are and the roles they play. So it's been found out that these cilia or hair-like structures are made up of microtubules that play a specific role in the paramecia. So now I've talked about the pellicle, I've talked about the cilia. Now the pellicle is kind of like the outer covering with the cilia coming out through it. Now these cilia are joined to basal bodies, which are known as the neuronemes. And these neuronemes are responsible for whatever the cilia does or whatever role the cilia plays in the paramecium. All right, so beneath the pellicle too, you can have what we call trichocytes. These trichocytes play two roles, either defense mechanisms or as well as anchorage. Do you get it? So that's the body morphology of the paramecium. Talking about the outer part, so you can have the trichocytes, the cilia. Yeah.
It's currently adding some people in the waiting room.
Okay.
Yeah.
Okay, I've done that.
Thank you.
Yeah. So I've talked about the outer parts, talked about the pellicle, the cilia, and then the trichocytes. So now, hello, your question. Is someone saying something? Does anybody have a question? If so, raise your hand so I can see you and call you. Okay. Yes, hello. Any questions?
Please, what was the color of the
color
cilia?
First of all, I told you that these organisms have a transparent body, so they basically wouldn't have a color per se. So it depends on the dye or the stain that you use in preparing the slide. That would lead to the color of the cilia that you see. And these are very small. You have the whole paramecium to be 0.22 millimeter. That's like a speck. So it will be very difficult to determine the color of the cilia. Noting because you can have one paramecium having about, let me say, probably like 5,000 cilia covering the body, right? So it'd be difficult to predict just what the color looks like. As I said before, they have a transparent body. So what the color of the stain you use preparing the microscopic slide would be the color you find, obviously. Is your question answered? The guy who asked the question, is he answered?
Yeah, thank you.
Okay, sure. So now, another notable thing of the paramecium is its nucleus. So if you look at the organism under a microscope, you'll find out that it has two nucleus, or two nuclei, let me say it that way, which is the meganuclei and then the micronuclei. Notably, is also its contractile vacuoles. It has two of those, one on the anterior end and one on the posterior end. So it is known, the contractile vacuoles are known to be star-shaped. Please note what I'm saying. I've mentioned the pellicle, I've mentioned the cilia, I've mentioned trichocytes, I've mentioned neuronemes. Then I've talked about the mega and micronuclei, and then I've talked about the contractile vacuoles. These are very notable things of the Paramecia, so you should take note. The contractile vacuoles are star-shaped. They are star-like in structure. And then, as usual, the Paramecia will have all other organelles. Why? Because it's eukaryotic, and eukaryotes have membrane-bound organelles. So you find, obviously, the nuclei, the mitochondria, etc. Everything, every subcellular mechanism you find in the cilia, but these are the very notable things that make you know what a paramecium actually looks like. Are we good? So talking about the cilia as well, there can be also two types. You can have the cilia that covers the entire body of the paramecium, which is called the body cilia, right? And then there's something we call an oral groove. So there are cilia that line up the oral groove as well. And then these cilia are also called oral cilia. So please, let's note that, okay, you can have two types of cilia, body and oral. You can have two types of nucleus, the mega and the micro. And then you can have two contractile vacuoles, which are the posterior and then the anterior. Are we clear?
Yes, sir.
Okay. So I'm moving on. So this is a picture of the Paramecium. Please, can we all see? Can we all see my screen?
Yes.
Okay.
Yes.
So as you can see, I talked about what the cilia, and as you can see my mouse moving, this is the cilia. The hair-like structures you can see all over are what we call the cilia, right? And then you have this big thing here is the macronucleus, and then the smaller one is the micronucleus. In some books, you see meganucleus. It's still correct because macro and mega obviously mean the same thing. But for the books that you're using in high school most of the time, they all use meganucleus. So it would be best to call this the meganucleus, and then the smaller one, the micronucleus. Then you have the star-shaped contractile vacuoles that I talked about. So you have one in the anterior and then the posterior ends. Can you see? Now,
Yes.
looking at the total organism, it looks like a slipper, right? That is the reason we say the Paramecium is slipper shaped. And then during my presentation, I talked about the fact that you can have cilia that line up the oral groove. This part I'm highlighting is the oral groove. So you can have a detailed diagram of the Paramecium, and you can find some cilia in there, and they have specific roles that they play. I'll talk about the roles very soon. Okay, so now I guess this is a more detailed diagram of the Paramecia. Hope you can all see.
Yes.
Yeah, so I've zoomed out a bit so that you can all see. So this is more detailed. Now take a look at the oral groove. You can find some hair-like structures in there. So that is what we call the oral cilia. This part, all of this part, these hair-like structures, they are the oral cilia. And then whatever you have on this side, all around, is the body cilia. Then you have the contractile vacuoles, you have the meganucleus, the micronucleus. Then these are food vacuoles. Then you can have your cytosol, your cystosome. Now, this cystosome plays a role, or cytosome plays a role like a stomach in the Paramecia. But obviously, because they are single-cell organisms or unicellular organisms, their mode of feeding is quite different, though they play the same roles. So yes, I'll give you like a minute, take a very good look at this diagram. Those of you that will take screenshots of it and note the drawing very well, because it's something that pops up most of the time in exams. You'll be asked to draw a Paramecium, or you'll have a Paramecia, you have a Paramecium diagram, and then you'll be asked to label some parts. So yeah, I'm giving you a minute, take a very good look at it. If anybody has any question concerning any parts, you can ask me, I'll answer. Then after a minute, I'll proceed.
Yeah.
Yeah.
Since when the meeting ends at 6, we'll use the same link for the carry meeting.
Okay, sure.
Yeah.
Okay. Yeah, David, you have your hand up. What's your question?
What are organelles?
The organelles refer to the subcellular components of the cell. So every other thing you find in the cell, like within the cell, or that lies within the cytoplasm, is what you call the organelle. So you can have your nucleus, you can have your contractile vacuoles, you can have your how do you call them? The mitochondria, you can have your Golgi body, you can have your endoplasmic reticulum. All those things are organelles. So those mechanisms that lie within the cytosol or the cytoplasm of a cell, which we will call the like subcellular components, are what we refer to as the organelles. Did you get it?
Okay.
Yeah. So note, you can see that in the diagram, the most tapered end, or the very blunt end, is what we call the posterior end. So if you have your slippers, I'll say the toe part of the slipper, this part is the posterior end, and then the rounded part is the anterior end. Okay, so I'm moving on. So now, for most of the organisms we'll be dealing with, it's quite a must that you know there are some things about them, right? So we are going to take a look at the nutrition. So which part of the Paramecia do you think will play a vital role with nutrition? Anybody? Anyone? You can just raise your hand up and mention one part that you think will play a vital role in the nutrition of the Paramecia. Oh, guys, no one is saying anything. Anybody? Just mention one part. Doesn't matter if you are wrong or right. If you are right, we'll talk. If you are wrong,
What's the
right.
question?
Mention a part of the paramecium that you think is vital
Of
for
what?
the part of the paramecium that is vital for its nutrition. Yes, Sandy. Sandy, your hand is up. Do you want to ask a question or you're
The
answering?
buccal cavity.
Okay,
I'm answering.
okay, you're right. What other part?
The food vacuum.
Okay, that's also correct. Anyone else? All right. Sandy, thank you very much for your contribution. So, because these organisms are single-celled in nature, they have some mechanism around them that helps them with their nutrition, which is quite different from how multicellular organisms feed, right? So now, remember when I was talking about the body morphology, I mentioned the fact that the Paramecia have oral cilia, right? So now, these cilia beat in a rhythmic motion or a rhythmical motion, right? And as they are beating or as they are moving, what happens is it creates some sort of wave that draws water that contains the food that it needs towards itself. So you have the cilia here beating in such a manner so as to draw the food to itself, but this food will be dissolved in water, or this food would be embedded in water. So the water that the food is in would be drawn into the oral groove, and it is caused by the beating of the oral cilia, right? So as you can see in my slide here, Paramecia mostly feed on bacteria. So if it finds the source of food, what it does is that it will anchor itself so that it doesn't move around, because it's single-celled. A serious gust of, say, water or wind or air can just push it away from the source of food. So once it finds its source of food, it anchors itself using the trichocytes at where the food is. Then what happens? It starts beating the oral groove cilia or even some body cilia so as to